In 2015, one of the most unusual and revolutionary experiments on human rejuvenation began. Volunteer and researcher Elizabeth Parrish agreed to change her own genome to stop the body's aging process. The results of the experiments surprised scientists and the public a lot. Elizabeth Parrish, a 50-year-old American researcher, was the first woman on Earth to have her genes artificially changed. The experiment was started in 2015, and a year later the first results were available to us. It sounds like the plot of a science fiction thriller, no less than Prometheus or Interstellar, but this is reality. Research on the aging process today is one of the most popular and promising. And not only because we all dream of being forever young, but also because, finally, scientific progress allows us to look a little deeper into the topic than before. A new brick in progress was laid by BioViva scientists, who in 2015 began their incredible experiment to change the human genome. Elizabeth Parrish, a volunteer and part-time one of the leaders of the scientific and medical company that started this experiment, was injected with genetic material that, having penetrated the nucleus of every cell in her body, was supposed to start changes that stop aging and rejuvenate the body. The results of such an intervention could be the most incredible. Firstly, no one has previously conducted such experiments on humans. Secondly, the drug itself is so innovative that it has not yet been approved by the U.S. Food and Drug Administration. So the injection itself was administered by Parrish in Colombia. Before that, she also had to record a video message in which she confirms that she understands all the possible consequences, accepts the fact that anything can happen, and also relieves others of all responsibility for what can happen to her and her body. The scientist also noted that even if the consequences turn out to be fatal, she still will not regret that she took such a risk, because any results of this experiment are incredibly valuable for millions of lives. In 2016, the first stage of the experiment was completed, which gave the first positive results, namely, the rejuvenation of Liz's body for 20 years was recorded. No, the girl did not look like a university graduate. The main changes occurred, of course, inside her body, which is much more important than elastic skin and shiny hair. According to new data, white blood cells in Parrish's blood became younger, and telomeres lengthened by 20 years. And this can be considered an incredible scientific breakthrough. It is known that the aging process is formed at the DNA level. It begins with a decrease in telomeres, the end sections of chromosomes. The length of these DNA sections is directly related to the biological age of a person. The older we are, the shorter they are. Cell division begins with the duplication of its chromosomes, thanks to an enzyme called DNA polymerase. Moving along the DNA chain, it synthesizes another identical chain. The problem is that DNA polymerase does not start its work from the very end of the chromosome, but slightly departs from the beginning of the DNA chain. Accordingly, with each cell division, part of the DNA simply falls out of the process. Telomeres are the very ends that are lost in the process of division. They do not contain valuable genetic information, so our body does not feel these losses until a certain point. However, sooner or later there comes a moment when the shortening of the chromosome ceases to occur due to these same telomeres, and then the DNA polymerase, starting its work once again, notices that the shortening begins not from an empty end, but from a segment with data. Then the enzyme can begin to connect different chromosomes to each other in order to repair the broken connection. With the accumulation of a certain amount of such compounds, the cell starts the mechanism of apoptosis, that is, the process of cell death. Simply aging. So, new data from the BioViva study suggests that the drug injected into Parrish's vein allows lengthening telomeres by 20 years, that is, delaying the process of apoptosis by almost a quarter of a century. And these are extremely encouraging results that can really turn our whole idea of old age and bring us closer to the dream of eternal youth. It is also interesting to note that despite the revolutionary nature of the study in which Parrish takes part, this is not the first attempt to lengthen human telomeres. In 2014, scientists at Stanford University developed a technology that also makes it possible to lengthen the ends of DNA. The drug they developed gradually increases the activity of telomerase, an enzyme that adds new sites to the end of DNA, for 1 to 2 days, during which telomeres rapidly lengthen. This technology was used exclusively on cultured cells, which are used to test new drugs and model various diseases. Of course, there was no talk of experiments on people at that time. The data from researchers at Stanford University, as well as those obtained by BioViva scientists, is incredibly valuable, as it proves that people are slowly getting closer to unraveling the mystery of the nature of aging. It seems that very soon we will learn, if not to program our genes, then at least to understand those processes that seemed a mystery for many centuries.